Okay, so here's our second part of the cellular respiration lecture. What we're looking at here is how heterotrophs, organisms that consume other organisms, get their energy. Mentioned earlier in the first part, there are two types of cellular respiration. One type is called aerobic respiration, which requires oxygen. The other one is anaerobic, which does not require oxygen. Both processes, though, are trying to produce ATP for the organism. The goal is to try to get as much ATP as possible. So let's consider an activity. Here's a triathlon. Triathlons involve swimming, biking, and running. So there's a massive expenditure of ATP that occurs during a triathlon. Short triathlon might take an hour and 10 minutes. A long one might take 12 hours. But while the individual athlete is exercising, their muscles are requiring large amounts of ATP. So they need to stay in the aerobic respiration pathway. They should have brought in a significant amount of energy in the form of carbohydrates. And then their body takes those carbohydrates, converts them into ATP through aerobic respiration. Now, if anybody has ever exercised, whether it's jogging, swimming, biking, running, played softball, soccer, baseball, etc., you want to stay in the aerobic conditions the entire time of your event. Because once you switch into anaerobic, your body is no longer very efficient. So athletes train their aerobic capacity. They try to increase it. So consider anybody who's ever been in a sport, the beginning of your season is all about building your endurance, increasing your aerobic capacity, so that way you do not shift over into anaerobic and shut down. So, like I said, with the swim, you might be out there for 10 minutes or an hour, but you need to maintain aerobic respiration or your muscles will fatigue and cramp up. While the athlete's on the bike, same thing. Continuously stay in the aerobic conditions. And then when you get into that run, if you don't stay aerobic, you're not going to make it to the finish line. So we have to stay aerobic, and we always want to make sure we have available energy going into the body, into the cells, to get converted into ATP. So we're going to explore aerobic respiration first, and then we'll go into anaerobic. Alright, so cellular respiration always begins with glycolysis. Glycolysis is going to be the conversion of glucose. So we're taking glucose and breaking it into two molecules that are called pyruvates. So literally you take the glucose molecule and break it in half. Now this occurs within the cytoplasm of the cell. The reason why we're working with glucose is it is small enough to actually get into your cell. It'll work its way, it'll diffuse through a cell protein, go into the cell, float around in the cytoplasm, and as it moves towards the mitochondria, it gets snapped in half. Now the things I want you to know about glycolysis: what goes in, what comes out. I am not expecting you guys to know every single step of this pathway. As you move on and take advanced courses in biology, you'll take a course in developmental biology, cell biology, then you're going to have to know every single step. So if you know what goes in, what comes out, for me, when you get to that advanced course, you don't need to, those things are already in your memory bank, you don't need to worry about those, you just need to worry about each step of the pathway. Okay, so to do glycolysis, you have to have glucose, and it costs 2 ATP. So there's an energy requirement to kickstart this reaction. So there's a -2 ATP requirement right here. You bring those 2 ATP in, and that begins breaking the glucose down. Now, as the glucose is broken down, bonds are broken, hydrogen gets released, and that hydrogen gets grabbed and attached to NAD. Now, if you remember one of the first slides, we talked about how NAD is essential for these pathways. Well, it's right here. I call NAD the little wheelbarrow, or the energy carrier that is going to grab the hydrogen and hold on to the hydrogen and create NADH. Okay, so we can think about NADH like this tiny little wheelbarrow. And that wheelbarrow is going to carry stuff. The stuff inside of it, that is going to be, that black stuff inside there, that is going to be the hydrogens. It's literally transporting hydrogens from the cytoplasm to the electron transport system. Now, the great thing about glycolysis, as the cell is going through this process, as the glucose is being broken down, it also releases energy, and there's enough energy given off to actually produce 4 ATP. So if you look down here, there's a production of 2 there and another 2 there. So think about this in terms of a business investment. It costs you 2 to start. You get 4 back. So you pay yourself back your original 2, and then you gained another pair. So you're up. You've paid yourself back and you've gained ATP. It's a great return on your investment. But you're also going to have these little wheelbarrows full of NADH. Now, the wheelbarrow parallel or concept, think about it like a stock. It's a stock or a bond or what we call a certificate of deposit, a CD. Right now it's not worth anything, but when you take it and cash it in later, that's when you get a great return on that investment there. Okay, so glycolysis starts the pathway. It's always the first step. Glucose gets converted into those 2 pyruvates, and this is going to occur within the cytoplasm of the cell. Now, if oxygen is present, if there's a large enough amount of oxygen present, the cell will go through cellular respiration and is going to run aerobic respiration. So specifically, it's looking at aerobic. Aerobic requires oxygen. So what we want to see now is if you're running aerobic, there's this conversion of the pyruvates. So the pyruvates, only when oxygen is present and we're doing aerobic, the pyruvates will get converted and moved within the mitochondrial space. Okay, so the conversion, they're moving through the mitochondrial membrane into the mitochondria during this conversion. It becomes a size issue. So the pyruvates are relatively large molecules. They used to be a glucose, but that got broken in half to pyruvates. And there is an enzyme called the CoA enzyme that comes in and attaches to the pyruvates, causes them to break apart. As they break apart, they produce 2 more of these NADHs. So now we have 2 more of those little wheelbarrow things we were talking about before. And we are going to convert those 2 pyruvates into molecules called acetyl-CoAs. Now the acetyl-CoA is going to be a smaller structure. It's smaller than the pyruvates, so it can continue to move into the mitochondria. That conversion will also release carbon dioxide. So carbon dioxide is given off, that diffuses out of the cell, into the blood, carried to the lungs, released into the atmosphere when we exhale. But what I do want you guys to focus on, during this pathway or this step, there is no ATP produced. But you got 1 wheelbarrow here, and let me put up the other one. You have a second wheelbarrow right over there. So as mentioned before, they're not worth anything right now, but these things carry hydrogens, and those are going to get used at the very end. So we had 2 NADHs produced from glycolysis. We now have 2 more produced from this acetyl-CoA conversion that is going to take place within the mitochondrial space. So reaction's starting to roll, and the key again is oxygen has to be present for these steps to continue. Glycolysis can happen without oxygen, but you don't do this one unless oxygen is present and it's available to get this pathway to go down the aerobic pathway. Okay, so now we're moving into the mitochondria. We're smaller. Our structure is tiny at this point, and we're going to move into the mitochondria in order for the next step of the pathway to occur, which is called the citric acid cycle, or you'll also see it's called the Krebs cycle. So the Krebs cycle is going to be occurring within the mitochondrial, what's called the matrix, kind of the center part of the mitochondria. So there's this big opening central part. I'll show you an example of it in just a second here. But the mitochondrial matrix is where the Krebs cycle or citric acid cycle occurs. Just like the previous step, I'm not worried about you guys knowing every single turn of this cycle. I want you to know what goes in, what comes out. Those are the key things for me. This is one of those things you'll need to know in greater detail as you move on. So if you get some of it down now, it'll make it much easier later on. Okay? All right, so the ingredient going in is acetyl-CoA. There's 2 of them because we had 2 pyruvates. Those got converted into an acetyl-CoA each. Each acetyl-CoA then goes into the Krebs cycle, and each of them goes through this cycle, Krebs or citric acid cycle. So as it goes in, the acetyl-CoA starts to turn, and the Krebs cycle spins it. And as it spins, it's being broken down, and pieces are getting thrown off of it. So when the hydrogens get knocked off of the acetyl-CoA, they wind up in the wheelbarrow here, the thing we call NADH. It's going to happen 3 times during the Krebs, or citric acid cycle. So we're going to get 3 more wheelbarrows per acetyl-CoA. So that's going to actually give us 6 total. The carbon breaks off and forms carbon dioxide twice. Once here, once here. Twice per acetyl-CoA. So we're going to actually get 4 things of carbon dioxide coming off. There is some ATP generated. There's 1 of them generated here, and that's 1 per acetyl-CoA. So that'll give us 2 ATP generated from the citric acid cycle. And then we have another carrier, this thing called FADH2. It's parallel to a wheelbarrow, but think about it like a trash can on wheels or some other concept. It's a carrier. Hydrogens go in it, and it has the ability to move the hydrogens over to the electron transport system. Okay, so all in all, when 1 acetyl-CoA goes into the citric acid cycle, it will give you 3 NADH, 2 carbon dioxides, 1 ATP, and 1 FADH2. So we have to look at it in an entire net yield here because there's 2 acetyl-CoAs. So it always goes in pairs. 2 acetyl-CoAs give us a total of 6 NADHs, 4 carbon dioxides, 2 ATPs, and 2 FADH2s from the citric acid cycle. Okay? So again, this is happening within the mitochondrial matrix. That is the space within the mitochondria.